Hi, my name is Tang, and today I'll be talking about how breast cancer can be treated using HDAC inhibitors. And essentially, first of all, we kind of want to talk about how breast cancer is important. As of 2024, it's the second leading cause of death in all women and the first leading cause of death in all women in less developed countries. Additionally, in recent years, breast cancer has been on the rise, highlighting the need to find a solution to such a pressing issue. So some general information about breast cancer itself. Breast cancer, like any cancer, is uncontrolled cell division, but in the breast. And it's classified by what receptors they lack, essentially. In the case of luminal A, it lacks the human epidermal growth receptor 2 or the HER2 receptor. In the case of luminal B, it in some cases lacks progesterone receptor. In the case of HER2, or human epidermal growth receptor 2 breast cancer, it lacks both the estrogen and progesterone receptors. And in the case of TNBC or triple negative breast cancer, it lacks all three receptors, meaning the progesterone receptor, estrogen receptor, and human epidermal growth receptor 2. And the reason why this is important is that without these important receptors, these cells can't respond to normal cues like normal cells would, which really allows breast cancer to be really impactful. So epigenome is really important to cancer, and this is because it impacts changes to gene expression outside of changes in the actual nucleotide sequence. And a really big player in the epigenome is acetylation, which is controlled by two different proteins. And it's HATs, or histone acetyltransferases, and HDACs, or histone deacetylases. And essentially how acetylation works is that when a histone is acetylated, the DNA is less tightly wound around the histone, which allows it to be more accessible, which allows for more gene expression. In contrast, when the histone is deacetylated, the DNA sticks closer to the histone, which makes the DNA less accessible, which therefore results in less gene expression. And essentially how acetylation works is that DNA is naturally negatively charged, while lysine is naturally positively charged. Lysine, by the way, is an amino acid found on the histone. Lysine is positively charged because of its R group, and since it's positively charged, DNA is negatively charged. There's this really natural attraction between the two of them. So when it's deacetylated, the DNA sticks close to the lysine. In contrast, when the acetylation mark is added by histone acetyltransferases, it actually negates the positive charge of the lysine, resulting in no attraction between the DNA, which is negatively charged, and the no longer positively charged lysine as a result of the negation from the acetylation, which results in the DNA not sticking as close to the histone, resulting in the DNA being more accessible, essentially. So next, we're going to talk a little bit about histone deacetylases. There are four different classes. Class 1 is HDAC 1, 2, 3, and 8. These Class 1 HDACs are generally expressed in most cell types, and they're all really similarly structured to one another. Class 2 is HDAC 4, 5, 6, 7, 9, and 10. These are generally not expressed in all cell types and are also really similarly structured to one another. In class 3 are the SIRCh-ones, which are the NAD plus dependent HDACs, in contrast to the zinc-dependent class 1, 2, 4. And class 4 is just HDAC 11, which essentially mostly serves immune functions. It wasn't similar structurally enough to class 1 or 2. So I wanted to mention that HDACs can control specific genes in some cases. And for example, in the case of HDAC1, it controls a specific growth regulatory gene called ERA. And essentially when HDAC1 is overexpressed, it will oversuppress this growth regulatory gene, which allows breast cancer to really take root. Another example would be in the case of HDAC11 and a specific tumor suppressor gene called aplasia RAS hormone 1. Once again, if HDAC11 is overexpressed, it oversuppresses this tumor suppressor gene, resulting in more aggressive breast cancer, essentially. And we kind of see a pattern here. Essentially, the HDACs will be overexpressed, which means that they oversuppress a specific gene that's important to combat cancer. And essentially, that's a really common pattern that we see when it comes to the importance of HDACs in cancer scenarios. Of course, there are some solutions that have been found for these problems in the form of HDAC inhibitors. First, there's class 1 selective HDAC inhibitors, which are selective towards class 1 HDACs. Some examples that I found were atinostat and trichostatin A. Atinostat has been shown to essentially turn estrogen receptor negative tumors into estrogen receptor positive tumors, which allows the letrozole treatment to be effective. I'll talk a little bit more about this later. And additionally, in the case of trichostatin A, it specifically combats the problem that I mentioned in an earlier slide based on HDAC1. And essentially, trichostatin A can return normal levels of ERA mRNA in the cell. Next are class 2 selective, which are selected towards class 2 HDACs. The example that I found for this one was VAS6, which selectively kills apoptotic resistant TNBC cells using their glycolytic metabolism alteration. And essentially, this specifically combats class 2 HDACs because class 2 HDACs, when overexpressed, have been known to create apoptotic resistant cells. Class 3 selective are selected towards class 3 HDACs or the sirtuins. The example that I found for this one was nicotinamide. Essentially, if you remember when I said that sirtuins were NAD plus dependent, so essentially this NAD plus is their substrate and the product is essentially their inhibitor, nicotinamide, which essentially inhibits through feedback inhibition, meaning that it may not last for a long duration. However, scientists have found that nicotinamide can also interestingly create conditions within the cells and tissues that stimulate the sirtuins, meaning that they've also hypothesized that maybe nicotinamide can be used as some sort of fine-tuning device for the sirtuins themselves, which I found really interesting. Next are class 4 selective or selective towards class 4 HDACs, which is specifically HDAC 11. The example that I found for this one was elevenistat, which is essentially just a hydroxamate-based HDAC inhibitor that selectively combats HDAC 11. Next are pan-HDAC inhibitors, which are non-specific inhibitors, meaning that they can have an impact on a large range of HDACs, essentially. The examples I found for this one were vorinostat and panobenostat, which have been shown to downregulate growth factors and survival proteins such as FOXO1, BCL2, while upregulating tumor suppressor factors such as P21 and P27. Next are chimeric compounds, which can essentially simultaneously modulate 2 or more targets. The example that I found for this one was pyrimethamine, which is both an antiparasitic while having control over the specific oncogenic transcription factor known as STAT3. STAT3 has been shown to increase proliferation and inhibit apoptosis or cell death. And essentially what chimeric compounds do is that they prevent drug-drug interactions, meaning 2 drugs having a bad reaction with each other within the body. And another really important problem with HDAC inhibitors is that they don't have an impact on solid tumors and hematological cancers, essentially. And so there's really been a growing need to find a chimeric compound that can both combat the solid tumors while having an impact on the epigenome. Something that I thought would be really important to mention is that some of these HDAC inhibitors haven't actually been tried in the human body. So some of these have been tried in like mouse models, cell lines, but some of them not in the human body. So some future steps that I see are deriving compounds from the pre-existing HDAC inhibitors. Specifically, there was a Chinese study published in 2021 on atinostat specifically, and essentially what it showed but that when atinostat was tried in the human body, it had many side effects such as neutropenia, thrombocytopenia, which means the lack of certain white blood cells and platelets. And essentially what driving compounds will do is minimize these side effects while increasing effectiveness. Additionally, I think that looking into combination treatments with HDAC inhibitors would also be really important. In fact, there has been a little bit of research into entinostat and the letrozole treatment. And as I mentioned before, entinostat essentially turns ER-negative tumors or estrogen receptor-negative tumors into estrogen receptor-positive tumors, which allows the letrozole treatment to be effective because the letrozole treatment relies on positive estrogen receptor status in order to lower estrogen and combat breast cancer. Thank you so much for watching my presentation. Have a nice day.